finally finished, right, chapter 4. We've been here for about a week now, looking at transport or energy. In the first part of the chapter, we talked about the kinetic versus potential energy. We talked about the first and second laws of thermodynamics. And we became familiar with, the oxidation reduction reaction. We also spend a lot of time studying the structure of ATP because we're going to see it used throughout the rest of the semester. First, starting here in chapter 5 coming up, so we'll use it quite a bit. And we also kind of summarize, well, what is energy necessary for? Then, where we finished yesterday, or Tuesday, is we are looking at what enzymes do. Okay? We learned that those are just biological catalysts that help enhance chemical reactions by binding to substrates at that enzyme's active site to produce products. And we didn't go into the mechanics, we just said, right, that's what an enzyme does. Okay? In addition to just understanding ATP and how it works inside of a cell, we also need to be familiar with how particles can be transported across a membrane. Okay? And in order for a molecule to be moved across a membrane, there has to be a gradient. What's a gradient? What? Very good. We have two good definitions. We can really simply define it with a slope, or in biology, we'll look at it as a difference in concentration of molecules in one spot compared to another. And so a gradient is just a difference in concentration of a molecule in one spot when compared to another. Again, a gradient is just a difference in concentration of a molecule in one spot versus another. Are you okay if I use the word concentration? I mean, more or less. Something can be more concentrated in one spot than the other. The spots, the locations that we're talking about are usually on either, right, the locations are either outside of the cell or the inside of the cell. It's usually where we're talking about. So we're talking about a membrane, right, separating two molecules when we look at concentration gradients at a living system. If there's a difference, think that difference spontaneously happened? No? It would have been, if there's a difference, it probably was, what? There's a difference in molecules between one spot versus another spot across the membrane. It was probably, eventually there may be some transferring, but if there's a difference, it was probably created. Okay? Because we know that most molecules try to reach what? Homeostasis, equilibrium. Okay? And so gradients are oftentimes created by a cell. Gradients are, at a living system, they're created. Extending that even more, if a cell creates a gradient, if a cell creates a difference, what would be the purpose of creating a difference? For different what? For functions, okay? The cell's going to create a gradient so it can use it for different fashions, right? Functions, whatever. The cells will create different molecular gradients so they can use them to do what? Moving molecules, right? We could start listing stuff here. Right? To do some form of work. if I use the word gradient, is that an okay word? Does that make sense? It'll be a word that I'll use frequently. So it just means the difference of a particular molecule in two different locations. It was probably created by the cell. And what we're going to describe is how molecules can move once there is a gradient established. And how molecules can move once a gradient is established is by what's called either active or transport mechanism. So molecules can be either moved actively or passively. In passive transport, we're going to see three subcategories. And they're listed here, and they're in the handout that you actually completed last week. The three types of passive transport include something called simple diffusion, facilitated diffusion, and another concept called osmosis. And those are the three subtypes of passive transport that we're going to study. What is characteristic of simple diffusion or facilitated diffusion or osmosis? What's common between all of those? Yes. Passive transport mechanisms do not require an input of energy. Okay, so that's characteristic right there. Passive transport does not require an input of energy. So for passively transporting a molecule, in a cell, we're transporting it across a membrane. If we're trying to transport a molecule across a membrane without using energy, which way do you think the molecule might be moving in respect to its gradient? What term could we use? What term could we use to describe the direction that a molecule might be moving and it's moving passively in respect to its gradient? Moving down. Very good. So passive transport, because it doesn't use energy, a molecule is moved down its concentration gradient from a greater to a what? Lesser area of concentration. We'll elaborate on that, but I just want you to understand the concept. Passive transport, does it use energy? No. The molecule is moving down its concentration gradient. It's trying to reach what? Trying to reach equilibrium. Keyword is trying. It may never reach equilibrium, but it's trying to. Yes. Let's contrast that to active transport. That name conjures up some images, hopefully. Active transport must use energy. So that's characteristic of active transport. Active transport does use energy. To move the molecule up or against its concentration gradient. reception is best describing what's called active membrane transport, but also we need to talk about the movement of vesicles. Do you remember the word vesicle? What's a vesicle? They're used to transport proteins from one organelle to another. Okay, they're just little membranes. To move the vesicle from, say, the rough ER to the Golgi to the cell membrane, that takes energy. So we're also going to talk about what's called vesicular transport, or moving vesicles. Got a couple of just simple illustrations. I'm going to first illustrate passive transport. The little red sphere is a molecule. It could be, say, oxygen, or a steroid hormone, or a diparticle, whatever. Okay, it's a molecule. I'm going to illustrate the concentration gradient by just drawing a slope. Okay. To be more accurate, I could have put an actual value of like 500 millimoles versus 1 millimole or something like that. Okay, but just trying to keep it simple for you. Also in there will be a membrane, because we're moving molecules across a membrane. Okay. So in passive transport, this molecule would move which way? Down the concentration gradient. It doesn't take any input of energy. It's just going to spontaneously happen in order for it to make that motion. Does that make sense? Again, do you love my awesome animation? I'm not drinking. What about active transport? If I want to move that molecule up against its gradient, I'm going to need energy. Our common form of energy is ATP. It's not the only one, but that's the one we're gonna pretty well study. Okay. So if I want to move that molecule uphill, I need to give it some energy, right, to push it uphill against its concentration gradient. Those are the broad differences between active versus passive transport. Does that not sound like something you might see on an essay question in about 4 weeks? Yes. Can you see where I go when I take time to walk through things now that you've kind of been through the test? Good. All right. What I want to now do is get more specific on each type of transport and look at the uniquenesses of each. And I've got this huge table, and it works for me, so hopefully it'll work for you. I've got each of the types. Passive transport in the orange, active transport highlighted in the pink, and then we're just going to go through, identify the categories that we talked about, the directionality, as well as give if there's any additional requirements. Okay, so the first one we're going to study is called simple diffusion. Okay, so simple diffusion, what type of transport is this? It's passive transport. So you know the direction. Write it down. What is it? Down the concentration gradient. Doesn't need energy. Here I said towards a lower concentration. You can say down the concentration gradient. It doesn't require any energy, but it needs what? It's implied when we're actually describing it. We need a what? Something to transport. We don't need anything to transport in simple diffusion. We need a, how do we define this? We need a unbalance. We need a gradient. Okay? So in requirements, we need a gradient. There has to be a gradient. There has to be that concentration gradient. So the particle can move down its concentration gradient. What makes up a cell membrane? What molecule makes up a cell membrane? Or you could say fat to keep it simple, right? And we know that fats, think about fats, okay, or oils or something like that. Best visual you can give is like you go to a restaurant and you get your oil and vinegar, you know how they separate, right? Okay. If I want to move something to that fatty layer, should it be water soluble? If I want to move something through fat, would something water soluble move through fat? What does fat do to water? Repels it. So could water or other polar molecules simply diffuse across the cell membrane? No. So if a particle is going to simply diffuse, it should be what? Water is polar, so water doesn't diffuse, so it should be non-polar. Okay, write this down. I know this is examples, but I want you to write down some clarification. If something is going to diffuse across the cell membrane, it better be non-polar. Not charged. Neutral. So non-polar substances can diffuse across the cell membrane much easier. Size-wise, what do you think a particle should be? Probably what? Small. So small, non-polar particles will simply diffuse across the membrane much easier if there's a gradient for them. So what we're adding is, if something's going to simply diffuse, it should be small and non-polar. Some examples of small non-polar substances. It says lipid-soluble substances that they're soluble in lipid. What you might say like steroid hormones. You've heard of estrogen, testosterone, progesterone, cortisol. Okay, those are examples. Water is listed there. It's one of those weird, in this class or in your biology classes, you can never say never or always. Okay, there's always an exception. Okay. Water is weird. So if you don't want to confuse yourself, don't write water down in this list. You can have it up here. It can do that, but it's odd. But what I'd like for you to add those other gases like oxygen. Oxygen, thankfully, is small and non-polar. It can just diffuse right across our cell membrane. That's good, right? Getting oxygen into a cell is good. Carbon dioxide can just simply diffuse which way? In your body, out of the cells. So small, non-polar particles will get across the membrane how? By simple diffusion. What questions do you have about simple diffusion at this point? Now, if we think about limiting the movement of molecules across the cell membrane of being small and nonpolar, that doesn't make sense because cells do need larger substances like, say, glucose or other proteins, to get across, or even water, okay, in large amounts to get across the cell membrane. Okay? And so some substances that are big or polar still need to get across the membrane, but we don't necessarily always need to use energy to move them. And so what can we do to get larger and or polar particles across the cell membrane passively? We need a transport protein, and the process is called facilitated diffusion. In facilitated diffusion, this is still a type of what? Passive transport. So the direction is still down, okay, towards the lower concentration for that particular molecule. What do we need? still need a what? Still need a gradient, but also a what? No energy, and a transporter. So in facilitated diffusion, we still need the concentration gradient, but now I have the word carrier, that's a transporter. Molecularly, though, the transporter is a what? What type of macromolecule are transporters? Protein. It's a protein carrier. But that protein carrier doesn't need any energy. All the protein carriers do is form a channel pushing out. Okay, so the pink thing that I've just drawn on the board is the protein. Little phospholipids. And can you notice how it's kind of like pushing out some space? So now the fluid on the outside of the cell is kind of connected to the fluid inside of the cell. So basically have a watery kind of passageway for something that's water soluble or large to kind of get through. So what kind of substances do you think we could transport thanks to facilitated diffusion? Substances that are larger or polar. I'm trying to think of something that's non-polar that's big. I can't think of a non-polar substance that is so biosynthetically large that it would not get into the cell by simple diffusion. And most everything else is charged, to be honest. Most everything else is polar. It's usually your lipid substances that are non-polar, that are like your steroids. They're just going to diffuse. You can't capture them. They're kind of free spirits, if you will, molecularly speaking. So at minimum polar. And then we've not talked about size of particles. I'm just going to use the word small versus larger. If you wanted to apply a value to that, when y'all take I think y'all may do a chemistry class sometimes too. I know at least one person's in chemistry. No. But Daltons, the molecular weight, so over like 100 Daltons is kind of the cutoff where something would diffuse versus need some type of facilitator. So to answer your question, most things are water soluble or polar. So I think I could almost easily, I hate to say always or never, but I could almost easily say most things that are going to be facilitated are probably at least charged at some point, like a little bit. Good question. Where we'll see facilitated diffusion in action is when we talk about chapter 6 in energetics. That molecule glucose that we've talked about some, that's a big substance. And we certainly don't want to spend energy getting it into a cell, but it's too big to simply diffuse in. And so we have things called glucose transporters, or you can call them glucose carriers. And they just bind to glucose and transport it from the outside to the inside of the cell. Doesn't require any energy. But why you're getting the glucose in is to fuse it, basically. And so some examples, other sugars besides just glucose, amino acids. Well, that's almost everything besides our lipids up there. of caution. When you start putting it together, a lot of times people will get inadvertently, because there's some similarities, facilitated diffusion and active membrane transport confused. So I'm gonna give you a heads up. Facilitated diffusion is a type of what? Passive transport. Doesn't require energy, but it does need a protein carrier, transporter. That doesn't need any. Do what? In the image that I drew on the board, yes, that would be like a protein folding to form a channel. Okay, so it pushes out those repelling, right, water repelling areas and opens up a channel. Just like a cul-de-sac would, like a, not a cul-de-sac, but a, wow. All right, so pipe, right, through what? Drain pipe or something, just like a drain pipe, and like you might find in your curb or something, let rainwater run off, okay? That'd be kind of like the protein. It forms a channel. It doesn't, like, in that case, the water doesn't have to be pumped from one spot to the other. It can just flow freely through. Same kind of thing with the glucose and other types of transporters. They just open up that space in the membrane so the molecule can just say, come on in, without having to fight through the lipids. So it's faster. Did that help or not? What do you mean? Yes, you're going a little more into biology than what we're gonna cover. They can be opened, they can be closed, but we're just gonna say for easy, they're always open. Molecule can just diffuse, facilitated into it. There are some different types that can be. They're called gated. Good. But so we'll just say they just let a particular molecule move which way? Down its concentration gradient without using energy. The types of molecules you might see facilitated properties, they are larger and probably polar. The other type of passive transport is called osmosis. And this is if you're going to get confused, this will be the one. Okay? But I'm going to try to present it to you so that it's not confusing. You probably joked about osmosis. You always hear somebody like, I hope to learn like the night for the test. I'm going to stick on my book so that I can learn by osmosis. You ever said that? Do I? It wasn't a cartoon. So, I haven't seen that. But it has nothing to do with learning. It has strictly to do with transport. And what type of transport? Passive. So movement of a molecule which way? Down. Here's what it is. It's so specific. It's movement of water which way? Down its gradient without using energy. But water's polar, so we probably need to have a carrier. They're called aquaporins. Aqua, water. Porin is a pore. Do it? A hole, okay, or a channel. So if we're trying to move water from one spot to another, we're still seeing a type of facilitated diffusion, but it's dealing specifically with water. Let me write this word down, aquaporin.
Osmosis really is facilitated diffusion.
Yes, and then
Specific type of within.
the reason it has its own category is because let me define it first. It's just, and I'll come back to your question. Okay. This is the passive transport of what? Which molecule? Water. Which way? Down its own concentration gradient. Water's charged, so it usually needs a carrier or aquaporin. So all that's comfortable, right? That's okay. So why don't we just call it facilitated diffusion? Okay? The reason it has its own category is because water can move across a membrane. A lot of times other substances that are charged, like say sodium or potassium ions, gate or a channel for it. Sodium has a gradient. It can't reach its own what? Equilibrium. And so water says, let me help you out. And it tries to dilute out more concentrated areas of solute. So this is where it gets confusing. So we're adding in there solute. We used the word solute the other day for first test, didn't we? Yes. In a solution is a mixture of a solute and a solvent. Here we're talking about water. So the solvent in our cells is water. Okay. So water can get across cell membranes easier using aquaporin. Okay. And most cells prefer for the water and solute concentration to be equal between the intracellular fluid and the extracellular fluid. I think you can handle that. Where we get into some situations are when the ion or the solute concentration is different. When the solute concentration is different, oftentimes these things can't simply diffuse, so they're trapped. So the cell says, it doesn't talk, but it says, acknowledges this difference and says, this isn't good. I need to dilute out where there's too much solute, and I need to concentrate where there's not so much solute, right? So where there's more water, I need to take some out and put it where there's less water. Okay? And that process is called osmosis. And so where this is different is because water's not just trying to reach equilibrium, it's actually trying to dilute out solute when the solute is not an equal concentration.
So when they are isotonic,
very good. We'll address isotonic, hypotonic, and hypertonic in just a moment, but you're right. In an isotonic solution, when the outside of the cell and the inside of the cell solute and water concentrations are the same, the ions may not be able to move. What happens is the water is moving back and forth so that there is no net gradient. Okay, so again, the ions may not be moving and may be trapped, but the water's done its work. Everything's hunky-dory. Homeostasis is set down.
so it's the same as the quantitative diffusion except it's the water only?
Yes. And that was the original question. And the yes, but, right, the big but is, the point is not to necessarily just reach water equilibrium, but to reach solute equilibrium. Okay, so that's why it gives its own little category. Because a lot of times the solutes are trapped in either the intracellular fluid or the extracellular fluid. They're trapped. And so the water can bring their concentration back to, hopefully, set point. So the direction is, and I want you to be real specific. What is water moving down its own concentration gradient? And I'm going to throw a wrench in here on purpose. Or you could say, no, that's the easy way to define osmosis. Water diffuses down its own concentration gradient. Or you could say water moves to the area of more solute. Water moves from where there's more water to less water. If there's an area of less water, it must have a high proportion of solute. So here, the water moves towards where there's less water, or you could say where there's more greater solute concentration. My brackets mean concentration. What's necessary? Okay, aquaporins and a gradient. I'll give some images in just a minute to help support this, but I just want to kind of go through and just contrast each of the different types of transport first. Okay. And so, it's water, okay? Just water moving. You can probably see this as an example. If you might get lettuce and keep it in the refrigerator till it's dying and you throw it out. You might do that vicious cycle. I do it. I pick it up, sit in your refrigerator, it wilts, you throw it out, and you go buy more, let it sit in your refrigerator, let it wilt, right? What you could do is put it in your fridge, it starts looking sad, put it in a bucket of what? Water. And water would move down this gradient from the bucket into the leaf cells. And the leaf will look alive and look good and yummy. So you could do a really simple osmosis experiment at home. You could put a leaf in pure water, or you could put a leaf in high concentration of salt water. What would happen to the lettuce in salt water? It's going to shrivel up because what just happened? Higher concentration of salt outside of the leaf cell drew the water out and causes the cell to shrivel up. That's osmosis. I'll put some images in a minute to help you. These are different types of passive transport. I want to contrast these two different modes of active transport and then follow up with some images. The first we're going to talk about is active membrane transport. And you know the direction is what? Up, or the word I use is against. But they imply the same thing. In active membrane transport, a molecule is being transported against its own concentration gradient to an area where there is more of that substance. If I want to move a substance against this gradient, we know what? We need energy. And also, what's going to use that energy and move those things? We need a transporter or a carrier. Yes. So we need a carrier and some form of energy. The difference the only other difference that we'll see in this class is we're going to use electrons. And then next week, and the rest of this chapter, we're going to see electrons. But for all other cell processes, ATP. And so a classic example of actively transporting substances is it's a creation of ion gradients, but other particles, nutrients, can be pumped against their gradient as well. In this class, we'll focus on ions being pumped against their gradient. Yes?
So you have sugars and amino acids under both facilitated and active membrane transport.
The difference is, if you remember, when we set up passive transport, we said there first has to be a gradient established. What establishes that gradient? Active transport. And if you remember, hopefully it'll...
Active membrane transport.
There's probably not going to be a difference, and there's no need for net diffusion, so oftentimes the cells will actually use energy to create a gradient so it can use it to do some other form of work. Okay? So classic example, you've heard of nerves before, right? What do they do? What do the nerves? They do what? Yes, and how? What's send messages, they activate, kind of spark, like electricity, right? Definitely electricity. What in our body can conduct electricity or carry electricity? It's on the list up there. Which one? That actually, no. And no. What has a full charge? What has a full? Ions. Okay. This is an example of why we would need active membrane transport. For example, in your nerves. We don't know how you guys don't know how they work, but we appreciate that they work, right? Okay? The way they work is they conduct electrical impulses. Or you could say they move ions across the membrane really quickly. Is what they do. Okay? In order to move them across the membrane quickly, there first has to be a gradient. And so your nerve cells will spend energy to create an ion gradient, and once that difference is established, it can, boom, facilitate them across the cell membrane fast to generate an electrical impulse. So where that was coming from is that cells use energy to create a gradient so that they can use it later to do some other form of work, and so, which does drive passive transport mechanisms. You'll have lots of examples in Chapter 5 and Chapter 6 putting these gradients into practice. But for right now, active transport uses energy to move a molecule up or against this gradient. We're going to need the gradient, and we're going to need energy. And we also need a what? A transporter, a carrier, something that physically moves that, whatever you're talking about across the membrane. Did I answer y'all's questions by chance? Right, so that's the active membrane transport. And there's also different types of vesicular transport, the formation of the vesicles. And that's what the rest of your chart will kind of walk you through. There are two primary categories of vesicular transport. One is called endocytosis, and the other is called exocytosis. Good. Endocytosis moves a molecule wrapped in a vesicle which way? Endo means into. Okay. And so in endocytosis, we're looking at a much bigger area of the cell, where the cell membrane will actually pinch around something and then encapsulate it, if you will, with a little vesicle. We had to trap something that was originally on the outside of the cell to enclose it, to bring it inside of the cell. Okay. So endocytosis brings vesicles into a cell. There are several subcategories of endocytosis. Okay, so let's review each of these. The first type of endocytosis is called, it is going to require, it's got the form of vesicle, sorry. The first form of endocytosis is called, you can call it phago or phagocytosis. Does anybody know about this one? Yes. Very good. Phagocytosis transports large molecules, or maybe even entire other cells, into the cell. So the key word that you'll want to hone in on phagocytosis is large. Does anybody know what the phago means? The prefix? Do what? It actually means, it translates to eating. It's referred to as cell eating. Okay, so it's referred to as cell eating. So you're taking in whole other cells or other larger components. The key word that I want you to focus on, though, is this large-scale endocytosis. could take stuff in on a large scale, or we could take stuff in on small scale. That is called Pinocytosis. So we're still just moving substances in. We're still forming a vesicle. So that's still common in there as well. But scale-wise, the substances coming in from the outside are, the vesicle being formed is really small. The Pino is referring to drink. So this is referred to as cell drinking. And that's how it can get larger amounts of fluids into the cell. Pinocytosis is the way that a cell can get extra fluids inside of it. It's not like, drinking out of a cup or something like that, but it's just pitching in sections of the membrane, taking in fluids, and kind of sample what's going on the outside of the cell. So the difference between phago versus pinocytosis is what? The size of the vesicle that's being formed.
Is it just taking in fluids, or is it taking in smaller molecules?
Yeah, if there's smaller molecules there, they're coming in as well. There's solutes and whatever's small out there would get trapped. So it's not just like, there's no sorting mechanism to say, no, just water come in. So it's going to sample everything that's small enough to
Small enough to get me.
So maybe ions and whatever else. Good question. A third type of endocytosis is called receptor-mediated endocytosis. So it still tells us it's endocytosis, but it needs what to activate it? A receptor. A receptor has to bind something, and that triggers this formation of a vesicle. So this one's unique in that a receptor, and I want you to write the word, I know I just have the word receptor here, but put and ligand, receptor and ligand, L-I-G-A-N-D, ligand. I'll write it on the board. Sorry. You can actually use my same image here. Instead of calling this a transporter, I can make it into a receptor. Do what? It's a receiver, okay, for a particular molecule. I'll make it fit in here. Kind of like remember our enzymes or our active sites. Okay, so the same kind of concept, except for when the molecule, the green triangle, binds to its receptor, what happens is that triggers this whole membrane to just engulf it. They just wrap it in and say, takes it in, and now that thing would no longer be exposed at the surface of the cell. It would be what? Inside, right? So I'm not going to draw that whole thing, but we're going to phase forward. Can I cheat and draw my membrane, the rest of my membrane like that? Okay. And the little molecule is taken in, and this would be not here. It just got pinched off and taken into the cell. And so that receptor receives something. It's endocytosis. It's receptor mediated.
How do you spell that again?
What? Oh, ligand. That's where I was going to write. Right. Ligand. L-I-G-A-N-D. That would be a term to describe whatever binds to the receptor. Okay, so we're adding ligand to the requirement. The receptor, and of course the ligand has to be present to activate the process. I could say ligand, I could say hormone if it's a hormone, some other chemical, a virus, or whatever it may be. And this is how big substances or macromolecules get into a cell when receptor-mediated endocytosis is used. Collectively, phagocytosis, pinocytosis, and receptor-mediated endocytosis are all forms of endocytosis. taking substances that were originally at the exterior of the cell, wrapping them in a membrane, and then secluding them into the cell. What questions do you have about endocytosis? Can cells expel their vesicles contents? Could a cell take a vesicle and then move its contents to the outside? Yes. What is that process called? Exocytosis. Exocytosis takes its vesicular contents and purges them out to the outside of the cell. In order to do this, the vesicle has to be formed, the contents, right, whatever's going to be secreted, has to be put together. This is how, you remember our rough ER manufactures proteins, the vesicles formed there, modified at the Golgi, and then packaged for release. Those vesicles that were modified, for example, at the rough ER, can get exocytosed out. So, for example, like your hormone, you've heard of insulin? It gets made like this, and it gets exocytosed out. The messengers in your brain called neurotransmitters, same thing. They're exocytosed out. So it's a way to get a large amount of particles, probably water-soluble, to get them out. The example out here is membrane proteins. You may remember from chapter 3, we noted that membranes have proteins embedded in them. Those got there by exocytosis. They got made in the cell and then pushed out, exocytosed, incorporated into the cell membrane. Questions about the difference between endocytosis versus exocytosis? Before I move on, these are under what type of transport? Active. Okay. I didn't address at all where the energy is used. Just when you take cell biology and you're a biology major, we'll tell you where that energy is used. Right now, it takes energy to form the vesicles and secrete, okay, and move. Okay. All right, so we'll go through some figures in your textbook. Far left, we have a particle, the blue particle, just diffusing across the membrane. What type of? Passive transport. No carrier, so this is simple diffusion. What type of molecule might simply diffuse across your cell membranes? Oxygen, my favorite. What else? What else might simply diffuse across a membrane? Carbon dioxide and other fats or lipids, like your steroid hormones, for example. Good. If anything is larger or maybe fully charged, we could still move it down its gradient, but we need to use a carrier. And what type of transport is that? Facilitated diffusion. But the commonality is you still see a gradient for that molecule. What questions do you have about the difference between simple versus facilitated diffusion? Osmosis, we learned, is a little bit confusing because it's dealing with the movement of which molecule? Water. Okay? And in order to move water, we grabbed an image that actually has the little transporters. But if we want to move water from where there's more water, the little red spheres, to where there is less water, aquaporins would be nice to have in this image. But if we want to move them right down the concentration gradient, try to reach equilibrium, where we said where it gets confusing is because water probably is moving because there's a difference in, not necessarily water, but a difference in solute. So water, yes, moves down its gradient. In other words, it moves to an area where there is more solute. And the whole point is to try to reach equilibrium. And this is some terms that we can kind of round out page 7 of your handout that Ms. Davis was leaning to a minute ago. If there is a difference in solute concentration across the cell membrane, the cell's not at equilibrium and we've got to address it. And so we can talk about a solution's tonicity. A solution's tonicity indicates if it can change the shape of a cell or not. If a solution's tonicity will indicate whether or not it can actually alter the shape of a cell by changing its fluid content. In our bodies, we prefer for solutions, okay, most notably the fluid outside of the cell versus the fluid at the inside of the cell, we prefer for the solutions to be what? Do we want a difference in water or solute total concentration? No. So in our bodies, we want solutions to be isotonic. What does the prefix iso mean? What? Anybody do any isotonic exercises at the gym? Do what? You could, I see where you're going, but and that muscle is doing what? The what? And that's where he was going, isolated. But iso is a change, a no change, steady. Okay, so if you're isolated, the numbers aren't changing. Isotonic muscle contractions is where you just hold a contraction. Isometric, same. Okay? Here we're talking about isotonic. Same what? And it will say the same, yes, but the tonic actually refers to the solute. So on the prefix for iso, that means what? Iso means same. Tonic means solute. Okay? Go ahead, Sonia. Okay. If the solute concentration is the same, what else is the same? In what part? If the solute's the same, not so far out. Yeah, you're right. But if the solute is the same in a solution, means solvent, which is what? Water. Okay. If the solute concentration is the same, the water concentration is the same, is there a gradient? No. In an isotonic solution, there is no gradient. There is no net what? Direction, flux of water. In an isotonic solution, because the solute solutions are equivalent, there's no gradient, and so water's not going to exit or enter more. It's just going to be equal. So the cell will stay balanced. If things aren't balanced, then they must be unbalanced. And so if we're looking at two solutions, they aren't equal. One would have more, one would have less. The area that has more solute, you would call what? Tonic. More solute, less what? Water. In a hypertonic solution, look, hang on. We'll go down, okay, to the next one. In a hypertonic solution, we said hyper means greater or more. The tonic talks about the solute. Okay? So if there's, what we're adding there is there's more solute, what you're saying, if there's more solute, there is less water. So if two solutions are not equal, water would flow always from which solution to which? Water will always flow from which of these to which? Which more or less, now listen, here's where we get, you guys, this is exactly where you get confused. If you just say more or less, are you talking about solute or solvent? You have to be specific. Are you talking about the solute or the water when you say more or less? Okay? So, let me restate that. If two solutions are not equivalent in tonicity, which fluid would water flow from? From the hypo, because it has more water, to the hypertonic, which happens to have more solute. Water will always flow from a more hypotonic environment to a more hypertonic environment to try to reach equilibrium. If it does or not depends on the condition, but that's the goal. In the lettuce, in the bowl example, the lettuce, this water inside of the environment inside of the cells would be considered to be what compared to the water in the bowl? Hypertonic compared to the water in the bowl, because that's just straight water. 100% water, no solutes if you're using pure water. So water moves into the plant cell. Now water is solute. Solvent. Dang it. Water is a solvent. The stuff inside of where you're going is the organelles, the nutrients, everything inside of the plant would be the solutes. Yes. Yes. So in animal tissues such as yourself, what do you think you prefer? Which? I actually prefer isotonic. Animal tissues, such as your body, prefer isotonic environments. No different. So that our cells don't swell or shrink. Plants, on the other side, though, actually prefer a more hypotonic environment. So they take in a lot of water and plump up and are able to kind of stand up in the environment. You may have it today. If you do, awesome. You have to review this before Tuesday because it'll just go as quickly as it came or faster than it came, okay? I want you to study that, and if you have questions, let me know. I want to use the next 5 minutes to go over that handout that I gave you guys on Tuesday because there are several questions. In your handout, I asked you guys to go through and look at basic chloroplast structure. And on Tuesday when we come back, we'll study photosynthesis, and we'll hone in predominantly or strictly on plants. But once we understand it in plants, we can apply it in other photosynthetic organisms. But we're going to focus on the structure of the chloroplast. Now I want you to go through and then identify the different regions. And so these were identified in the book, but the function wasn't put out there because that's a little bit probably more than we need to go to. Okay, so I'm going to work from the outside in. Collectively, this is a chloroplast. Where my hand is the outer membrane. There's a space, so you could call it intermembrane, and then the inner membrane. There's an outer membrane, there's an inner membrane, and there's a liquidy environment. What is a liquidy environment inside of stroma? Where we'll spend our time is in these little green discs. What are those discs called? here's where you guys are confused on the handout. I asked you the function of the outer and the inner membranes. Okay, for each of those, just write compartmentalization. They're there to compartmentalize the reactions of photosynthesis. Compartmentalize to contain. On the outer and the inner membranes, for us, we're just saying, well, for both of them, let's just say they're there to compartmentalize. In addition, I want you to acknowledge that not only do they compartmentalize, but they transport substances into the chloroplast and out. And so they compartmentalize and transport. Yes, because the question was, do they both? Yes, because this is inside of a cell cytosol. These things are making what for us? Well, there's DNA in there, but for the plant, they're really making energy in the form of sugars. Okay? And so they're making sugars, but we got to get the product out. Okay? But we got to get the precursors in. So where I wanted you to acknowledge is that the outer and the inner membranes not only compartmentalize, but they transport. It should make sense coming off the heels of chapter 4. You're going to get molecules across the membrane. Light bulb. Good. I like light bulb. The other question that was on the handout was, what the heck does the word lumen mean? The word lumen just means the inside of something that's hollow. Okay? So in this image, it's the dark spaces in the thylakoids. And I think the book must use the word thylakoid space. I could talk about the lumen of your stomach. I could talk about the lumen of a female's uterus. I could talk about the lumen of your bladder. What's the difference between lining? Lumen is just the void space. A lining would be something that creates a surface, I guess.
I still don't understand what it means.
Lumen is just the opening. So if you were to look at your, for example, right, your Red Bull's going to be our weekly example. If you were to cut your Red Bull in half, I could say the inside of the can, or I could say the lumen of the can. The lumen holds the drink. Okay? The lumen of the thylakoid is where the reactions occur that we're going to study. I think those are the primary questions. What other questions do you guys have on that? What are the granum? Grana. A granum is multiple thylakoids bound together. So on Tuesday, you're submitting that handout. Add as much detail as you can. Because as identified, photosynthesis is really difficult. And so the more proactive you are this weekend, the less painful Tuesday and Thursday will be.
You're not wanting to turn it in again.
Correct. They are due on Tuesday. See you guys. Have a good weekend.